So let's get going. So good evening, everyone, and welcome to our public virtual meeting on cyanobacteria blooms. Why do they happen? What are the risks? And what can we do? This is a virtual public meeting that's being put on jointly by the Vermont Department of Environmental Conservation, Department of Health, and the Lake Champlain Committee. Next slide. So just to briefly go over the meeting agenda, I'll start with some welcoming notes and meeting ground rules. The second section will be a presentation on cyanobacteria ecology and context. We'll then hear about some of the health issues associated with cyanobacteria blooms. From there, we'll go into a brief presentation on how to recognize cyanobacteria, which is just a teaser for a lot more detail that can be provided there, but it'll be enough to get folks started. We'll from there discuss what to do if you see a bloom in regards to how to avoid it and how to report it to the state. And we'll nearly wrap up with a presentation on what DEC is doing to reduce blooms in the state. And then we mean to leave about a half an hour or more at the end for question and answer. So that's our agenda for this evening. Next slide. So just who our speakers are, my name is Oliver Pearson, and I'm the Lakes and Ponds Program Manager for Vermont Department of Environmental Conservation. And we have a very qualified and expert group of speakers who will also be presenting tonight. And I'll let them introduce themselves in the order with which they'll be speaking to you this evening. Peter, did you want to just go back and introduce them? Can we just introduce ourselves at the start, and then I'll go on to the next slide?
Sorry, I misunderstood. Yeah, hi, I'm Peter Isles. I'm an aquatic ecologist with Vermont DEC Division of Lakes and Ponds.
Hi, everybody. I'm Sarah Vos. I'm the state toxicologist with the health department here in Vermont.
Lori, you just need to unmute yourself. Sorry.
Sorry. Hi, everyone. I'm Lori Fisher. I'm Executive Director of the Lake Champlain Committee. We run the Cyanobacteria Monitoring Program and train community science volunteers and coordinate that with our partners, the Vermont Department of Environmental Conservation and Vermont Department of Health, as well as New York Health and Environmental agencies.
And good evening, everybody. I am Bridget O'Brien. I'm the Senior Toxicological and Radiological Analyst at the Health Department within the Environmental Health Division there.
Great, thanks everyone. So we have some of the state's leading experts in cyanobacteria that'll be speaking to you this evening, and it's a great group. Next slide, please. Just want to once again welcome everyone and thank you for joining us again. Just go over some of the virtual meeting ground rules. As I said earlier, the meeting is being recorded, and the recording will be shared after. We'll put it up on the Agency of Natural Resources YouTube channel. So if you want to see this again, or if you want to share this with your neighbors, that'll be available, and the link for that will be up on one of our social media pages, so you can find it. Please keep your microphones muted during the meeting unless you're speaking during the Q&A session. That just helps reduce background noise and feedback. As I said, we aim to have at least 30 minutes of Q&A at the end of the meeting. So to ask a question, there's two options. There is a chat box at the top of your screen. There's a little circle with like a little call out circle with some lines in it. Click that. That's the chat, and you can type in the question if you prefer. Or when we start the Q&A session, you can use the raise hand feature in Teams. It's also up in the top of your screen. There's a little face with a hand, and if you click on that, a little menu comes down and there's an actual hand. You can raise a hand, and that way we'll know that you'd like to ask a question. And if you prefer to go that route and ask a question verbally, don't forget to mute yourself. Please identify yourself as you begin your question or comment, and try to speak slowly. I struggle with that all the time. So we will attempt to address all the comments or questions that we get in the chat box and with raised hands. If we do run out of time and there's questions in the chat that we haven't gotten to, we'll try to address those in writing after the meeting. The This is a public meeting, so please be concise and respectful of others, of course. And if you're having bandwidth issues, you can turn off your video, and sometimes that helps, which I'll be doing in a second here. So with that, I'm going to hand it over to my colleague Peter Isles to begin the technical content of this presentation. Peter, over to you.
Thanks, Oliver. And yeah, thanks everybody for coming tonight. It's great to see a good turnout. So I'm going to start off giving a bit of background on the ecology of cyanobacteria and some sort of recent trends in broader context. As I said earlier, my name is Peter Isles, and I'm an aquatic ecologist with the Vermont DEC. And my specialty is really cyanobacteria and other lake processes. So I just wanted to start off with the basics. What are cyanobacteria? Basically, cyanobacteria are unicellular organisms, although they sometimes live in colonies of many cells, and they're capable of photosynthesis. So they're actually the first organisms on the planet that develop photosynthesis, and as such, we can thank them for things like having oxygen that we can breathe. Cyanobacteria live in almost every environment on Earth, and they are a natural and an important part of most ecosystems. So I say this because, it's not always a cause for concern if there's a little bit of cyanobacteria present in your lake, because, there are cyanobacteria present in every lake. Where they start to cause problems, though, is when habitats are altered. Situations can arise where cyanobacteria grow too much, and when they reach very high densities, which we call blooms, that can cause a variety of problems. Which we'll go into. So what do these problematic cyanobacteria in Vermont look like? Under a microscope, this is what sort of 5 of the more common, potentially dangerous cyanobacteria look like. And you can see that there's a variety of shapes. You have things like microcystis, which is kind of blobby, covered in mucus, where you have these long filaments of Oscillatoria or Phanosomnon. Delicospermum, I think, is quite pretty. It has this kind of beads on a string morphology. And all these different growth forms correspond to different life strategies and different habitat preferences. So there might be certain lakes more prone to bloom of one species or another. And of course, that can mean you have different toxins present or sort of different times of the year that you need to be more wary of blooms. So some common features of all these species is that they tend to form large colonies. That most of them can regulate their buoyancy, which means they have little, basically gas bubbles inside them, which allow them to float up and down. And this can cause these accumulations of surface gums with very high densities under the right weather conditions. And of course, they can all sometimes produce toxins, and you'll hear more about that in the next presentation. So that's what they look like under the microscope. What they look like sort of at the large scale. Here are some images of some of the larger cyanobacteria blooms in the world. So I'm sure a lot of people are familiar with Lake Erie. The next picture is the Baltic Sea. So this is a huge area covered with a bloom of nodularia. Taihu Lake in China has sort of perennial microcystis blooms every year. This is one of the largest natural lakes in China. And more recently, along the Florida coast, we've seen really a major increase in cyanobacteria blooms there. And that's even in salt water, which is sort of not as common an occurrence. Usually cyanobacteria are the dominant bloom-forming species in freshwaters, but saltwaters you often have other groups. And of course, Lake Champlain. Luckily, Lake Champlain is not as bad as these other systems, but we do have some problem areas where we do get recurrent blooms, like you see Missisquoi Bay in the north here, which is quite green. So what exactly are blooms? It's not really a clear definition, so you can be excused if you're not quite sure when you see a bit of green in your lake, if that's a bloom or not. But in general, for health purposes, we can say that blooms are times when there are high concentration of cyanobacteria, when the water looks green, and you'll see some pictures of what the water actually looks like in one of the later presentations from Lori. And cyanobacteria blooms, as I said, can cause several problems, and these include the direct effects of the toxins on humans, pets, and wildlife, but they also include sort of ecosystem consequences like oxygen depletion in fish kills and changes to the structure of lake food webs that can be quite wide-ranging. So that's kind of what cyanobacteria are. Now I'm going to get a bit into what actually causes cyanobacteria blooms. So before blooms can happen, cyanobacteria need the resources to grow. And you can think about this as kind of like a recipe. I find that useful. So if you're going to make cookies, you generally start with some flour, sugar, eggs, whatever. You put them in the oven at a certain temperature for a certain amount of time, and you have your cookies. You can come up with a recipe for cyanobacteria as well. But here the basic ingredients would be things like carbon, nitrogen, phosphorus, and light. Again, you need to incubate them at the right temperature for the right amount of time, and you get a bloom. So in this recipe, the things that we humans can control are really the additions of nitrogen and phosphorus, and in particular phosphorus. These are things that tend to come from human activities largely, things like agriculture, urban land uses, lawn fertilizer, or sewage runoff. And phosphorus is a key thing to control in the case of controlling cyanobacteria blooms because it's often the nutrient in short supply relative to the recipe. So again, going back to the analogy, if you had sort of all the flour in the world and all the sugar in the world, but only 2 eggs, then you couldn't make that many cookies. And if you went down to 1 egg, then you could only make half as many cookies. So in this analogy, the eggs would be the limiting nutrient, and in this case, phosphorus is the limiting nutrient in our lakes. That's why we target so many efforts at reducing phosphorus in all the lakes in Vermont. So because of all this, blooms are most common in lakes where we have increased nutrients from human activities. And these nutrients may occur now, or they may be nutrient additions that occurred in the past, but those nutrients take a long time to leave because they're stored in lake sediments and in the soils in the surrounding catchment. So often, once you take action to reduce nutrient inputs, it can still be 10 or 20 years before you see major improvements in lake water quality. Right, so one question I think that a lot of people wonder about is, are blooms getting worse? And I think it's clear to say that there's more stories about blooms in the media. There's more public awareness of blooms, but it's not always clear, whether this reflects an increasing trend in blooms or just increasing sort of interest. And I think the answer is probably a bit of both. So if you look at some studies of sort of long-term trends in blooms, this one is looking at really long-term over about 200 years using sediment cores from all the lakes where these dots are on the maps. And anywhere you see green, you see a lake where cyanobacteria have increased. So the basic takeaway is that over this sort of long-term timescale, cyanobacteria have increased, across at least North America and Europe, and I think this pattern holds up elsewhere as well. However, if you look at kind of a more short term, so this data set just goes from 1984 to now, or to 2012 using satellite data, you see a much more mixed picture, particularly in areas like North America. You see some lakes with increasing trends in cyanobacteria shown in red and pink, some with decreasing trends shown in the blue colors. So the takeaway is that over the long term, we've seen increases, but I think once people became aware of the importance of phosphorus pollution and started taking action to control it, you started to see blooms decline in more places. To bring it to a Vermont scale, I think we can say that it's not really clear if there are any trends in Vermont based on the data that we have to date. We do have a lot of reports of when cyanobacteria blooms occur, and these come from the Cyanobacteria Monitoring Program that we run in DEC together with the Lake Champlain Committee and the Vermont Department of Health. So these are, on the right side of this dashed line, these are all visual assessments that volunteers and staff send in saying there's a bloom here or there's not a bloom here. And you can see that we get more reports over time, again, reflecting that increased interest in blooms. But we don't see any clear indication that there's a sort of higher proportion of alert level reports, and those would be the yellow and the red categories. So it's not to say that there is no increase, but it's to say that this data set is probably not good enough to give a definitive answer. So that's Lake Champlain, and we have even less data from other Vermont lakes, but we are starting to get more. So all you're looking at here is Vermont inland lakes where we get reports of cyanobacteria status. And we're up to something above 35. So this is great, but as you can see, these data don't go back very far, so we can't say much about long-term trends. I will also say that not all of these lakes have blooms. These are just lakes where we have people keeping an eye out for blooms. Some have never actually reported a bloom. But if you'd like to participate in this program, you're very welcome to get involved. You can find us on the website and then reach out and say, I think we should monitor on my lake, and we would love to have you. And last, I just wanted to touch a little bit on what we should expect in the future from blooms. And this is an important question because most research agrees that climate change is likely to make blooms worse. This is because of several reasons. The first is that warmer water is just more favorable for cyanobacteria growth, so they can grow faster. But also, there's sort of indirect consequences like longer summers, which allow longer periods of thermal stratification, which can lead to more internal loading, more internal release of phosphorus from sediments, and stronger blooms. And finally, most climate models predict that there will be more storms and precipitation in the Vermont area, so more nutrients coming into our lakes. So this is all the bad news. But the good news is that most research also suggests that nutrient management and human actions are much more important than climate in controlling blooms. So this means that we have the potential to control blooms even if warming continues by managing nutrients now. And the uncertainties associated with climate also suggest that we should continue monitoring the situation so that we can recognize any increases in blooms as early as possible and take action. So I will leave it there. And as Oliver said, we will take all the questions at the end, but I would be happy to answer anything you post then. And now I'll turn it over to Sarah to talk a bit about health issues associated with blooms.
Okay, thank you. Can everyone see my first slide, cyanobacteria and health effects?
Yep.
Great. Okay. So thanks, Peter. That was a great introduction to cyanobacteria and had a lot of good information for us. So I'm just going to spend a few minutes talking about cyanobacteria and some of the possible health outcomes and give you an understanding of why we take cyanobacteria blooms really seriously here. So the first reason, as Peter mentioned in his presentation, is that cyanobacteria blooms can produce toxins. And I've listed the 3 main toxins that we monitor for here in Vermont. Those are microcystins, Lindospermopsin, and Anatoxin. As Peter pointed out, some species are able to produce 1 or 2 types of toxins. Some can produce lots. We never really know when a bloom is producing toxins, and that's something that scientists are still trying to understand. Even when they have the genetic material and the capability to produce toxins, sometimes they do and sometimes they don't. One of the reasons that we are concerned about cyanobacteria is that there have been several reports of dog deaths after dogs have been swimming in the lake or recreating in ponds in Vermont. So we know that these blooms have the potential to be harmful. We also know that children can be more sensitive to cyanobacteria blooms than adults, and that's for a couple reasons. You'll often see, especially very little children like toddlers, playing in the very shallow water, and in many cases that's where blooms can accumulate, so they may be exposed to them more. But also children have a higher exposure per body weight to these toxic chemicals. And that's something that's true for almost every chemical exposure, is that children get a higher dose than adults. And then the last reason that we're concerned about cyanobacteria blooms is that swimming and recreating in these blooms is the primary route of exposure. That's the way that we know that people are most likely to get sick from cyanobacteria. And of course, with so many beautiful lakes and ponds here in Vermont, people are recreating all summer. So we know that there's a high potential for people to be in these areas where there can be cyanobacteria. There have been a couple of media stories, and these are just 2 of many that have been published in the past several years. In some locations around the world, the cyanobacteria blooms and the outbreaks have made people very, very sick. So the picture on the left is a picture of a lake in Utah. I think this was in 2016. So the lake was completely covered in a cyanobacteria bloom, and over 100 people reached out to the Poison Control Center because they were very sick from it. So they had symptoms such as vomiting, diarrhea, headaches, and rashes. So that, the large number of people getting sick was attributed to people being at the lake and swimming in that water. There's also a case report. This was in 2017, which is one of the more recent case reports. This was in Uruguay, so again, not here in Vermont, but it was a case where a family, 3 adults and 1 child, were playing in a lake that was having a very severe cyanobacteria bloom. The whole family got very sick. They were vomiting, diarrhea, stomach cramps, etc. The adults recovered very within a matter of a day, but the little child, who was just a toddler, ended up having liver failure. So again, just to, this isn't the situation here in Vermont, but this is one of the reasons that we keep a close eye on cyanobacteria and monitor what kind of toxins they're making. So I'll just give a little more information about the toxins and what we know about their toxicity. So microcystin is the most common toxin that's produced worldwide. So when you hear us talk about toxin testing, we often use microcystin as our routine. We monitor for that one the most because that's the one that we see the most here in Vermont, and it's also the one that's most common around the world. We know that microcystin primarily targets the liver, and there's a pretty specific reason for that. It's because the liver produces these little peptides on the outside of the cells, and I think of them as a bouncer. So these peptides are kind of the gateway to get inside the liver. And so when the microcystin gets in our bodies, these peptides, or the bouncer says, "Hey, come in here, you belong here." And so they actually transport the microcystin into the liver. So that explains why we see liver toxicity across the board in almost every kind of mammal that's exposed to microcystin. We also know that the kidney and the gastrointestinal system are affected. That explains some of the other symptoms that people experience after being in a cyanobacteria bloom. There's also some information about reproductive system issues. There's some indications that microcystin can result in lesions in reproductive organs and decrease sperm motility. But overall, the primary concern is for liver toxicity. Cylindrus rhamopsin is not as well studied as microcystin, but it is one of the ones that we've just started testing for in the past couple of years. It's not found as frequently in Vermont as microcystin, and it does also target the liver and kidney, so some of the health effects can be a bit overlapping. And many blooms will produce not only microcystin, but they can also produce Cylindrus rhamopsin and other toxins. And the last one that we monitor for is Anatoxin, and this is a different type of toxin. The first 2 target the liver. This one targets the nervous system, specifically the brain. When it was first identified, it was named very fast death factor, and there have been some recent publications that have highlighted that sort of nickname. But the reason why it's so toxic is that it's a cholinergic agonist. So it binds to our neurotransmitter system. So in the brain, our neurons send out little chemicals that make our body breathe. So that's a really important function. It makes our muscles contract and relax. And of course, our diaphragm muscle is one of the most important muscles that we need to contract and relax. So anatoxin interferes with that signaling in the brain. And because it binds so tightly, our body's natural clearing system, the natural way that we can make our muscles relax, doesn't work if anatoxin is bound there to that neuronal cell. So this can result in the overstimulation of muscle cells because they keep contracting and they don't know how to relax, which will just end in fatigue and paralysis of the muscles. So when you think about paralysis of the diaphragm, that can be very dangerous, and that this may explain some of the dog deaths that happened here in Vermont. So this is just a summary slide that gives an overview of some of the other symptoms that have been reported. It's a different way. I think Peter did a nice job presenting the different types of species, that the cyanobacteria species that can make these toxins. So we do know that these species are here in Vermont. Generally speaking, though, we don't detect very high levels of toxins. We sample blooms when we are able and can get to them. So we do sample quite a few. We try to get the thickest, scummiest part of the bloom to see how high the toxins are. And you know, again, we don't really see very high concentrations of toxins, which is very good. But we're always keeping our eye out to see, you know, if blooms start changing. We also do a lot of monitoring of clear water, and I think others will probably talk about this as well. In some parts of the world, we've seen that when the blooms dissipate, the clear water left behind can have high toxin concentrations. So far, we haven't seen that here in Vermont. So there's one other area of research that I want to talk about, which there's a group of researchers who are looking into the link or a possible link between cyanobacteria and neurodegenerative diseases, specifically ALS. So there's a chemical, which is a non-essential amino acid called beta-N-methylaminoalanine. We'll call it BMAA for short. BMAA is made by some cyanobacteria, and there are some researchers who are studying a theory on whether that BMAA is associated with ALS, which is amyotrophic lateral sclerosis. That's a neurodegenerative disease. So ALS is unfortunately a disease in which about 90% of cases don't have an explanation. So only about 5 to 10% are genetic. There are some proposed risk factors such as smoking, things like head injuries, so people who play professional sports and have many head injuries. And then some occupations may lead to a greater risk for developing ALS, especially working with metals and pesticides. But because 90% of ALS cases are unexplained and ALS is such a devastating disease, it is important that this research, you know, all research into the causes of ALS continue. The BMAA hypothesis to date, the existing data do not show that exposure to cyanobacteria and BMAA cause neurological diseases, including ALS. There's 2 links here, and we'll make sure that the PDF becomes, and these links become available, but there are some 2 websites if you want to get more into the science and read some more about BMAA. I would point people to those websites. Another question that we get a lot is about aerosolization of cyanobacteria or aerosolization of particles. So can the cyanobacteria lift up from the lake and travel miles to people's homes and expose them there? So the phenomenon of aerosolization does exist, and this schematic gives a nice visual summary of how that can happen. So we know that things like jet skiing, there are some other recreational activities that those can produce those sprays. So those do have the potential to go up into the air. If there are toxins there in the water, then the toxins could be included in those droplets. When people inhale those droplets, the most commonly reported symptoms are irritation, so irritation to the eyes, nose, throat, and lungs. There are some researchers who are looking to see how far those droplets can move. But so far, the most likely route of exposure, the way that most people are most likely to get sick from cyanobacteria is being in the bloom, in the water, recreating in it. So, our current advice, which, you'll hear this from Bridget and Lori, is to stay out of the water if there's a bloom. So that's protecting people from the known risks of cyanobacteria. And I think this is my last slide, but I just want to make a point about drinking water. Peter showed you some pictures of Lake Erie. Back in 2014, the public water system, which you can see a picture there on the right, that sample is taken right in front of their water treatment system. So the public drinking water was pulling in raw water from this very thick cyanobacteria bloom, and the finished drinking water had microcystin in it. So that led to 400,000 people being on bottled water for several days. So that after that happened, which of course was a very national big news story, we started offering weekly cyanotoxin testing to the Lake Champlain public water suppliers. at no cost to them, because we recognize that while a lot of the public water system inputs are very deep, and our lake doesn't have the types and the, you saw from Peter's slide that most of the visual observations are green, we still wanted to get a picture of our weekly drinking water. Since 2015, we haven't had toxins in our finished drinking water. We've only had a couple of very, very low, right at the detection level, detections in microcystin that were never confirmed on the next sample. And the last note about water is private drinking water. So people who pull their private drinking water from a surface water source should have a treatment train installed to remove cyanotoxins, as well as other pathogens. It's really important that untreated surface water not be used for drinking. We've also gotten some questions from people who have drilled wells, maybe next to a pond or close to a river that experience cyanobacteria blooms. And generally speaking, those deeper drilled wells are in different aquifers and they're not susceptible to cyanobacteria contamination. Okay, so that's my last slide. And again, I'll take questions at the end and I'll turn it over to Lori.
Thank you, Sarah and Peter and Oliver. Hello, everyone. Thank you for being here. I'm Lori Fisher. I'm Executive Director of the Lake Champlain Committee, and I'm here to give you a teaser to how to recognize cyanobacteria. We run a host of training sessions during the summer season as well as year-round on this, and they take a lot longer than 10 minutes. So this is a whirlwind tour, and we'll link you up to some resources where you can learn more. And you're welcome to take an educational training from us later in the season. First slide here is showing cyanobacteria blooms on Lake Champlain. And you can see this white and turquoise color here. Those are indications of the bloom breaking down. And so if it were producing cyanotoxins, they would be released into the water column. As Peter and Sarah have noted, cyanobacteria don't always produce cyanotoxins. We don't know what causes them to turn on that switch. So we have a focus in our approach of teaching people how to recognize it, avoid it, and also report it. If you see scummy water, if you see something that you think is cyanobacteria in the water, you want to be cautious, stay out, avoid contact. So why are we so focused on cyanobacteria? Has a lot of positive uses, and it's a positive attribute and an important part of the ecosystem. But as Peter was noting, when it gets out of hand, it can be a cause for concern. And the trends are something where we're just seeing more potential for blooms. So similar to the situation with ticks, when 20 years ago, if you lived in this region, you were an outdoorsy person, you could hike, roll down a hillside, not have to do a tick check. That's no longer the case. And it's similar for cyanobacteria. If you're a water person, you now need to know how to recognize it, avoid that contact, and hopefully report it so we can keep everybody safe. So a few tools to help you do that: gloves, a jar, and a stick. Okay, these, and I'll walk you through why. You want to avoid having contact with anything. A jar is going to help you take a water sample. Cyanobacteria do different things in the water, and you can think of your jar as sort of a microcosm for what's happening in the water. It'll help you give a sense of what's going on. And then a stick is helpful because some cyanobacteria, some things that are confused with cyanobacteria, you can pick up with a stick. Cyanobacteria, you generally can't. So before I talk about cyanobacteria and show you some more pictures, I want to go into phenomena that are often confused with cyanobacteria. Top three are duckweed, filamentous green algae, and pollen. Duckweed is this tiny, free-floating aquatic flowering plant. If you look up close, it looks a lot like little lily pads, but from a distance, it can often present like cyanobacteria, particularly that pea soup green color. One way to help differentiate it, if you see this from a distance, cyanobacteria is generally going to have a more fluid edge, not this like table-like presentation where it just ends. As you get closer, you'll be able to see that leaf-like structure, that plant matter. Cyanobacteria doesn't present like that. It's not a plant. It doesn't have that plant leafy structure. And when you're seeing cyanobacteria, you're not seeing anything individual. You're seeing these microorganisms coming together once they're visible to the eye. So again, getting up close to duckweed, you can see, even in this fuzzy photo, the individual leaves, the thallus of the duckweed cyanobacteria. It's not like that, even though the color may be confusing. Filamentous green algae, we often get a lot of reports of people confusing filamentous green algae with cyanobacteria because it can form, these scums. It's often presented in colors that aren't dissimilar to how cyanobacteria can show up, but its texture is usually stringy or hairy or silky or clumpy. It often will attach to rocks or plant or woody debris. Most cyanobacteria don't do that. And then most filamentous green algae you can pick up with a stick. Most cyanobacteria you can't. So do the stick test. It's a good way to help you right off the bat differentiate from green algae. And here you can see this filamentous green algae draping off this stick almost like fabric. Pollen, very prevalent now. It comes in shades somewhat similar to cyanobacteria, but usually a lot brighter. And it's going to show up on the surface water, and you're generally seeing it, though, at different time than you would see cyanobacteria. So when you're thinking about how to differentiate pollen from cyanobacteria, think about color. Pine pollen in particular is a much brighter yellow than cyanobacteria usually presents. Think about seasonality. Cyanobacteria generally show up, are much more common midsummer to late summer, certainly early fall, after the water's warmed, whereas pollen is more of a seasonal attribute in the spring and early summer. You also want to think about the surroundings. When you see pollen, you're usually going to see it on the water surface, but also collecting along the shoreline and in the parking lot. Look, if there is a nearby parking lot and cars have been there for a while, look on the windshields, etc. Look at the surrounding area. Cyanobacteria is more going to be relegated just to the surface water and maybe, depending upon wind and wave action, showing up a bit on the shore. Pollen will be in a much broader area generally. So when you start to see cyanobacteria, and when it first becomes visible to the human eye, it often shows up as little fuzzy balls or looking like glitter or confetti. You see them showing up in the water, not too dense a fashion here. And similarly here, jar test is helpful because if you take a jar test, okay, you glove your hands and you invert your jar and move it through the water column and then carefully put it down in the sun for about 15 or 30 minutes. If there's cyanobacteria there, it's going to rise to the top. It's going to move. Remember, Peter noted, many cyanobacteria have those gas bubbles, so it's going to rise to the top towards heat and light, doing in your jar just what it's doing in the water body. Plant matter is going to settle down to the bottom as with sediment. So that's why you want to give it about a half an hour. Don't shake this up too much, because if you do, it's like giving it a little blender action, a whirlwind of activity, and it's going to be slower to do its thing. You also can see it in the water there, again, as these tiny dots, okay? This is, again, the beginning. It's not a bloom. Blooms are when they're all coming together in more of a colony presenting with surface scums. Now it's getting denser. This is what we call a low alert bloom for those of us who run the monitoring program. Later on, someone will be talking about the cyanobacteria data tracker. These kind of conditions show up on the cyanobacteria data tracker as a yellow dot once they're reported. You might see streaks along the shoreline and in the water body and all these different striations, but a low alert condition is also going to be more restricted in its area. One of the differences between a low alert and a high alert bloom is extent. And here you're seeing narrow along the shoreline, clear water beyond. So when you see it in a jar, less density, but after a half an hour, collecting here at the top, again, some streaks, striations, but you're not seeing open water appear green or blue-green or like a paint spill, okay? So that again, restricted to nearer areas, may show up as patches smaller than a softball, narrow band along the shoreline. And again here, more restricted in its area, clear water beyond, low alert bloom. Now we've moved into full high alert bloom, looking like a paint spill. This is pea soup green, classic color, but it doesn't always present that way. This is one that's easy to call because looks like a paint spill, it's got that density, and the colors here are more representative of that green continuing. So this is also, all these are high alert blooms in progress. This again is showing cyanobacteria breaking down. The signal for that are these blue and turquoise colors. And again, if cyanotoxins were present, they would have been being released into the water column. This is showing surface scums coming in this yellow-green color, and then here showing a lot of density along the shoreline and that tinge of green throughout the water. Even when it's not as dense, it's showing that color. If you do the jar test, greater density here after about a half an hour. There's so much density that, you know, they're all fighting to get to the top. Surface scum on the water, wide band along the shoreline or in the water. And the open water appears green or blue-green, you know, has the same tinge of color. So here again, high alert bloom. This is showing up on St. Albans Bay, south of St. Albans Bay, the Georgia shore. It's a public drinking water intake. I think Bridget will cover that in a moment, and concerns with cyanobacteria. And here's a late season bloom. This was October 30th at Little Eagle Bay, Rock Point in Burlington. So October 30th in 2019, blooms can happen still later in the season. That's why we want to always, even when the weather's not, you know, the water may appear too cold for you to go in. That's not the case for your pets. So you always want to be checking the water anytime you're strolling along it with little kids or pets, anybody that is potentially going to have contact with it. Again, cyanobacteria breaking down, that turquoise and white, clear indications of that, this heavy density throughout, not seeing the bottom of the water. And that ends that whirlwind tour. I just want to note our contact here and places where you can get more information. We produce a weekly cyanobacteria email throughout our monitoring season. You can sign up for it on our website. You can also take a training to get more information. And also, very importantly, check out the cyanobacteria data tracker that's housed on the Vermont Department of Health website. It will include all the reports from inland waterways and Lake Champlain once they're vetted by LCC and our partners who are here tonight. It's a terrific resource. You can click on any dot and get information on that report. And then also for bloom conditions, you'll see pictures. So it'll help educate you more about how to recognize cyanobacteria and avoid it. So thank you very much. I'm going to escape here and then try and move out of this. Thank you.
Hello, everybody. Hopefully you're seeing that my slides. All right. All right.
Yep.
So again, I'm Bridget O'Brien, and I work at the health department as a cyanobacteria lead. And thanks for that great intro on what cyanobacteria blooms look like, Lori. Now that we know that, we're going to move kind of into what to do if you see a bloom. So the first thing I wanted to mention that we want everybody to get in the habit of scanning the water for cyanobacteria blooms before swimming, which might not be something that everybody is doing currently. And then in addition to looking at the main water body where you're planning to go swimming, don't forget if there's a beachy area to look at the small puddles and pooling along the beach because cyanobacteria can grow in those little puddles and kids might play in those, dogs might drink from them. And then we didn't quite get into too much in in Lori's presentation, but there are also cyanobacteria that are called benthic cyanobacteria. And that means that they usually grow on the bottom of water bodies, but they can break off and wash ashore. And this is also stuff that you can pick up with a stick. And it is suspected that across the US, dogs have become ill and died after eating chunks of these benthic cyanobacteria pieces. And kids would also, have an interest in putting that in their mouth and eating it as well. So you're going to want to be on the lookout for those kinds of things and try to keep your pets and kids from eating things along the shoreline that might be benthic cyanobacteria mats. So if after you're scanning the water, you see a bloom, we want you to avoid recreating in that water. It's best not to swim, wade, or boat in that water with cyanobacteria blooms. As Sarah mentioned initially, the ingestion of cyanobacteria, so swallowing cyanobacteria and the toxins, is the biggest known exposure pathway for health effects. So we strongly recommend avoiding exposures where you might swallow the water. And especially, again, kids and pets are more likely to experience those health effects. So we want to make sure that they're staying out of the water if you see a bloom as well. If you're at a managed beach, we want you to tell the person who's in charge of running the beach, that could be the lifeguard or the manager, so that they can take measures to ensure that other people aren't exposed. That could be posting signs to alert people or closing the beach. And then finally, we would love for you to report the bloom to the health department by filling out the form on our website that you can see here. There's just a bit of information that we collect on where the bloom was, what it looked like. And then we also really want you to include at least 3 photos of the bloom from different angles and different distances so we can see what you're seeing. And the multiple photos help counter the difficulties of photographing water, including glare and reflections. And we'll talk about what happens with those reports in just a minute. But first, I wanted to mention that if you think that you have been in contact with cyanobacteria, if you realize that you were out swimming and then you came back in and you swam through a bloom, we advise that you rinse off with clean water as soon as possible to try to get the toxins and cyanobacteria off of you. And then if you think that you have symptoms after having swum through a bloom, we recommend that you talk to your healthcare provider. They're going to be able to provide the best advice on what to do, how to address any symptoms you have. But we would also like you to report those symptoms that may be related to cyanobacteria exposure to the health department as well. So going back to reporting the blooms to the health department, all of the bloom reports that come in from the public as well as from the trained monitors are reviewed by partners at the health department, the Department of Environmental Conservation, and the Lake Champlain Committee. And so we're looking at the report, the description you provided, and the photos that you provided, most importantly. And if the photos show that a cyanobacteria bloom or the start of one was happening, the report is added to this online cyanobacteria tracker map as either a low alert or a high alert, depending on the size and density. In this way, anybody with internet access can see that a cyanobacteria bloom was reported there. It includes the report status, where it was, what the water body was, what town it's in, and then, as Lori mentioned, we have those attachments here that you can actually see the photos that were submitted with the report, which has been a really great educational tool. so that others can learn what cyanobacteria blooms look like, especially in that area where you just reported it. And this tracker system has proven a great tool for people looking to see where cyanobacteria blooms have recently been reported. But we want everybody to remember that the information is only as good as the most recent report. And because conditions can change so rapidly with cyanobacteria, everybody really needs to learn to recognize and avoid cyanobacteria, so that even if you see a green dot on the map, when you're going there, you're still scanning the water and looking for those blooms just in case. We're also always looking for more monitors, so if you're interested in adding data to this tool, please reach out. And you're going to hear that probably more times even in this this presentation, as we're all really interested in adding more monitors. We encourage all lake associations and towns with water bodies to work together to develop a cyanobacteria monitoring program for your recreational water areas. And this will really encourage a quick response for cyanobacteria blooms. Partners on this call can provide guidance and training, as we've noted. And this cyanobacteria guidance for communities document that's on our website will also be helpful in that. And that's not to, replace, we still want that information going on our tracker map, but it might just, make sure that your local response is consistent, everybody is in the know in that community where you are. And then I briefly wanted to touch on some of these other exposures aside from swimming exposures. So Sarah mentioned drinking water and what happened in Toledo. This slide is a little more geared towards private drinking water sources, where we really recommend not drinking untreated surface waters and working with a water treatment specialist to make sure that your treatment system for your surface water system will remove toxins, and then testing your drinking water for toxins through the public health lab if there has been a bloom near your intake, just to make sure. For boating, again, Sarah kind of mentioned this, that boating can lead to toxins in the air through the droplets that might be produced. So it's not recommended that you're boating and jet skiing through cyanobacteria blooms, as you're more likely to breathe in those water droplets. Regarding eating fish from cyanobacteria blooms, it's not recommended to eat fish caught in blooms. There's ongoing research. We don't quite know how long cyanotoxins may remain in fish organs when they swim in the blooms, but we do always recommend cleaning fish properly, not eating the organs, especially the liver, where cyanobacteria toxins may accumulate. And then I just kind of wanted to close on our key takeaway message that I really hope you leave with today, and that is when in doubt, it's best to stay out. It's not worth risking it. So if you think you're seeing cyanobacteria, it's not worth swimming. So when in doubt, stay out. But that is my last slide. So I think you're up, Oliver.
Great. Thanks, Bridget. And hopefully folks can see a slide here with some information about what Vermont DC is doing to reduce blooms. We're a bit behind schedule, but this is the last section of the presentation, and then we'll get to question and answers. Thanks for your patience so far. And this will be a very, very whirlwind tour of what some of Vermont Department of Environmental Conservation is doing to reduce the frequency and magnitude of cyanobacteria blooms in our public waters. And so, starting from some of the key federal and state statute about our approach to clean water, we recognize that the clean water supports fishing, swimming, boating, and other uses such as aquatic habitat, bolsters tourism, and helps to maintain property values and provides access to drinking water. And we're trying to maintain that clean water at good to very good to excellent standards across the state. And so what the Vermont DC does is coordinates across other state agencies and the local and federal partners to develop and implement projects that improve water quality. That's our basic point of departure. So we've heard, from other speakers that phosphorus pollution contributes to cyanobacteria. That's often a limiting nutrient for algae and plant growth. in our waters, and phosphorus can lead to other water quality issues. And so one of our principal tools to reduce phosphorus pollution to our waters and thereby reduce cyanobacteria blooms is to develop clean water restoration plans. You may have heard the term TMDL, or total maximum daily load, which essentially is a plan that identifies what are the pollutant reductions required for an impaired water to meet the state standards. So Lake Champlain, Lake Memphremagog, Lake Carmi, those are some examples of waters that don't meet our good standard for clean water in the state. And so we've designated them as impaired. And we've developed these total maximum daily load plans to turn that around and restore those waters to good, very good, or excellent status as under our Vermont clean water, our Vermont water quality standards. And so these plans, these TMDLs, they target non-point source and point sources of pollution, and identify projects and efforts that can be implemented across the landscape to reduce those sources of pollution. And they also include loading reduction targets. So how much do we need to reduce this external loading of phosphorus to restore these water bodies? And we work over time to achieve those targets. And DEC also uses tactical basin plans, with the states divided up into about 15 different basins. And those plans at that scale identify actions necessary to achieve our pollutant reduction targets. And just recently, the Vermont Lakes and Ponds Program, working with other partners, has begun working at a smaller scale, at the scale of actual lakes and their watersheds, to identify the stressors to those lakes and identify also projects to mitigate those stressors. And so we're starting to use this Lake Watershed Action Plan as a local level tool. So what we've heard, to put this, to make this a bit more concrete, we've heard, summarizing what other speakers have said, that excess nutrients, calm water and warm water can combine to lead to cyanobacteria blooms. We can't do much at the scale of our lakes about calm water and warm water. We know that's perhaps getting exacerbated because of climate change. So we really focus on this excess nutrients part of Peter had that great analogy of a recipe for blooms. We focus on how do we reduce those excess nutrients. And so this is a nice graphic that shows here in the center what a cyanobacteria looks like in a body of water. And over here on this side, you see some of the sources of those nutrients that we're trying to reduce. And so more common in Vermont is non-point sources like agricultural runoff. We have a bit of urban runoff, or runoff from impervious surfaces, particularly when there's storms and lots of precipitation and water flowing down streets and into our water bodies. We are also managing wastewater effluent. And so our efforts to implement clean water projects are focused on those sources of nutrients that can lead to cyanobacteria blooms. And so, what we've done is we've divided up the state at that scale into 3 total maximum daily load regions, and we've developed phosphorus reduction plans for each of those regions. So up here on the left on the map is the Champlain Basin, and that has 1 TMDL focused on phosphorus this year. In the middle northern part is the Lake Memphremagog, which also has a TMDL, and then the whole Connecticut River side of the state, which drains into Long Island Sound, also has a TMDL, but that's more focused on nitrogen and less relevant for our discussion. And finally, as I mentioned, Lake Carmi, which is up here in Franklin County, is a lake that suffers from excessive blooms, and that has its own smaller scale TMDL. And so these plans target phosphorus pollution to limit cyanobacteria blooms and other challenges. And we can see here that for Lake Champlain, with land cover, while 73% of the Lake Champlain Basin is covered by forests, and only 18% is agriculture and 4% is developed land, that 18% agriculture leads to 38% of the phosphorus load to Lake Champlain, and the developed land leads to about 16%, and with the other sources coming from forestry, stream banks, and wastewater treatment. So we're really focusing a lot of our TMDL efforts on reducing non-point sources of phosphorus from agricultural lands, and then using different tools to reduce stormwater runoff. So one of the key tools to reduce phosphorus pollution to our lakes is to implement voluntary clean water projects, which target nutrient and sediment pollution across different land use sectors. And so these projects are sometimes required by the TMDL, which is a federally approved, EPA approved plan. The Vermont Clean Water Act has its own requirements, but they're often voluntary projects as well. In terms of resources, over the last 5 years, Vermont has invested $194 million into clean water projects, and with those funds, we've leveraged another $70 million in local and federal sources. So we are spending significant resources to improve water quality in Vermont, in the Champlain Basin, in the Memphis-Magog watershed, and in other parts of the state. So some examples of the projects that we've implemented: agricultural BMPs have been put in place on 90,000 acres of agricultural lands. We've improved 200 miles of municipal roads to reduce stormwater runoff from those roads. 332 acres of impervious surface, think parking lots, at some of our larger shops have been treated for stormwater. And there's a lot of restoration going on of riparian areas on streams as well as lake shorelines to reduce erosion and runoff from impervious surface in those areas. And then a lot of passive restoration, so putting land into conservation. And for those of you that live on the shorelines of lakes, there's also the Lakewise Program, which is our program that helps property owners near lakes implement lake-friendly practices. A few other things that the state has put in place to try to reduce cyanobacteria blooms a number of years back, because of some real problems with blooms at Lake Carmi. The Vermont legislature passed the Lakes in Crisis statute, which first has a 3-part test to define when a lake is in crisis, and Lake Carmi did indeed pass that test. And then that program it allocates funding annually to address this lake in crisis and restore the lake. We have implemented in very few cases in-lake treatments to try to reduce cyanobacteria blooms. In general, our approach is to try to address the source of the problem, which is the phosphorus loading to the lakes, and most of our interventions are therefore on the landscape and in the watersheds of our water bodies to try to reduce the external loading of phosphorus. But in a few cases, we've implemented in-lake treatments to either deprive blooms of the nutrients that they need, and that's what can be done through an alum treatment. The alum binds with the phosphorus, makes it inert or unusable, and prevents it from being available for plant growth. That's worked well on a couple occasions, but really for an alum treatment to be a good investment, you have to first get a handle on reducing the external loading of phosphorus into the water body, and so it only works in certain cases. And in Lake Carmi, we knew there that low oxygen conditions at the bottom of the lake were leading to phosphorus release from sediment at the lake bottom, and so we put in an aeration system to ensure that the lake was oxygenated and mixed, and to prevent that sediment phosphorus release. That was a very expensive investment. It requires annual maintenance and operating costs, and does have some negative environmental impacts. So that's not something that we would do very frequently. It's sort of a reserved for very unique situations. And finally, I would be remiss if I didn't mention the regulatory aspect of our work to reduce phosphorus pollution to our water bodies, and a few examples of how regulations work hand in hand with our efforts to improve water quality are here. The 3-acre stormwater permit to reduce stormwater runoff from impervious surface, ensuring that wastewater is managed properly at 92 facilities across the state, and our efforts to reduce combined sewage overflows, and then protection of different key water resources across the state: wetlands, lakes, and stream banks and floodplains. So we also do a lot of monitoring, and that helps us understand where the problems are and how to focus our mitigation efforts. So our monitoring on Lake Champlain and in its tributaries helps us identify what are the annual loads of phosphorus coming into the lake. And load is basically the concentration of phosphorus times the amount of flow, the volume of water coming into the lake. And with that, we can calculate loads. And what this map is showing our annual loads from a few years back. And you can see that the tributaries to Lake Champlain that had the highest loads into the lake were the Missisquoi and the Winooski. And so in those rivers watersheds, that's where we focus a lot of our efforts to reduce phosphorus loading. And we also look at phosphorus concentrations in the lake, and to see where are areas with the highest concentrations, and try to focus projects in those areas as well. And so you can see, as Peter mentioned, Missisquoi Bay and St. Albans Bay up in the northeast arm of the lake are the areas with some of the highest phosphorus concentrations, and we're working specifically in those areas to try to reduce loading and bring those concentrations down over time. Briefly, we have some smaller lake-specific efforts as well. This is a photograph of a buoy we've deployed in Lake Carmi, which allows us to monitor lake conditions. This buoy gives us basically real-time data to look at temperature, dissolved oxygen, which is important so we can ensure that the lake bottom is not becoming anoxic or deprived of oxygen, which can lead to sediment phosphorus release. And then this bottom measure here, which is a measure of phycocyanin, which is a pigment in blue-green algae or cyanobacteria, and that really helps us see is a bloom occurring or not. Which this very boring straight line at the bottom is a good thing. That means there aren't so far this year we haven't seen any blooms in Lake Carmi. Let's knock on wood that situation continues. So this is just an example of how we monitor lakes. So last slide, what are the results that we're seeing? We are seeing some progress, which is great. For example, in the Champlain TMDL, over the last few years, we've reduced 28 metric tons of total phosphorus load to the lake, which represents about 13% of our overall target that we're trying to achieve by the year 2038. So we've gotten going on that. Of course, a lot more work left to do. Similarly, in Lake Carmi, there our interventions in the landscape and the watershed and in the lake have reduced about 251 kilograms of phosphorus. That was just in 1 year. And our total progress there is about 41% of our overall phosphorus reduction targets to meet the Carmi phosphorus TMDL target. And we've made some similar progress up at Lake Memphremagog, and we continue to monitor those results. So in conclusion, we are making a bit of progress. We'll continue to use clean water resources, both state, federal, local funds, to achieve more results in terms of improving water quality. Lots more work to do, and I think cyanobacteria blooms will be a part of Vermont's freshwater ecosystems, but our goal is to reduce their magnitude and frequency through projects and regulatory work. So that's the end of our formal presentation. Just want to thank everyone for their patience and sticking with us for the last hour. And what we have the five of us are now available to take any questions that you in the audience have. So we've gotten to hopefully the interactive part of our session tonight. And so what we'll do is we'll take a look at what we have in the chat, and if any questions have come in there and they haven't been answered yet. I think one or two questions have come in, but we've tried to answer those. So let's go over to our participants and see if anyone has their hand raised and would like to ask a question verbally. And what I'll do is I'll call on folks. Please unmute yourself and ask your question, and then one of the five of us will do our best to answer it. So far I see that Tom Spencer has a question. So go ahead, Tom.
Hi, it's actually Carol Spencer. I just wonder, in your very good presentation, that last statistic about 13% of the phosphorus reduction in Lake Champlain as of 2020, how many years has that been, and what have you found to be the most effective ways of reaching the agricultural community in order to get that reduction?
Thanks. I'll take that question. That's from 2016 to 2020, so that represents the first 5 years of the implementation of the Champlain TMDL, and so we have from 2020 to 2038. It's another 18 years to get from 13% to 100%. So lots more work to do. To the second part of your question, working, as I said, I think that the statistic was, it was either 90,000 or 900,000 acres of farmland that had been on the receiving ends of best management practices. It was 90,000 to reduce, phosphorus nutrient pollution coming from these non-point sources, agricultural inputs across the state. What's been effective? I think we're seeing a number of different interventions that are effective, and one sort of point of departure for that is where we can provide funding and technical assistance to incentivize our agricultural community to implement these types of projects. That's a great point of departure. From there specifically, I think using cover crops so that fields are not harvested and left bare, which makes them more susceptible to stormwater runoff, has been effective. Sort of manure management, helping farmers with installing and better manure pits and then managing those ensuring they don't fill up and become, that they can be covered. So if there's a rain event, they don't fill up. That's been effective in reducing sort of the whole linkage between manure and phosphorus pollution. One thing that we've started only in a few watersheds around the state, but has great potential, is rather than spreading manure, with a spreader on land, is manure injection. So using a sort of a specialized piece of equipment to inject the manure into the soil that achieves that, soil fertility benefit, manure disposal goal that farmers are trying to achieve, but doesn't lead to that manure scattered on the top of the fields where it's more susceptible to runoff. So those are a few examples. I think ditching is something, managing ditches that are linked with agricultural fields, so that those don't erode in stormwater events is important. And then, I think there's some general, approaches to conservation agriculture and fertilizer minimization that have been used at farms across the state that have shown some promising results. So this is a little bit outside my area of expertise, so I think I'll stop there and avoid misspeaking. But if any of my colleagues want to contribute to answering your question, I'll give them the floor. I think looking at the, if you want more information about that question, the Vermont Clean Water Progress Report from 2020 goes into a bit more detail about the specific agricultural interventions and BMPs that we've been supporting with Clean Water Fund, that the Agency of Agriculture has been supporting as well, and that might give you a bit more detail if you're interested.
Thank you.
Great. Any other questions?
I see a couple in the chat there.
Okay, great.
I can take a stab at that first one. The first one is from John H, and that was, can you elaborate on what kind of water treatment system is best for treating lake water for drinking? We have a 3-stage filter system, which includes an ultraviolet stage. Is this sufficient? And I would say that we would definitely recommend you talk to a water treatment specialist, but the ultraviolet is unlikely to help with any toxins that would break through, as that would generally kill the cells but not treat toxins. So it would, kill E. coli, but not treat toxins, which are the chemical then. But you can check out our drinking water webpage for private drinking water on the health department's website. And there is a link there that gives some advice on how to find a water treatment specialist. So I'd recommend you check that out. And I don't know if Sarah, anybody else has anything else to add there?
Yeah, I just posted, it's a pretty long set of rules, but DEC does have a potable water supply rule. So that went into effect in 2019, and that does describe in detail the types of treatment trains. I'm not an expert in it, and I just opened it. I don't even know if I can explain it. But I'll also just put in the website for that DEC program, so you can, I don't know, Oliver, if you know of a specific person to contact, you could offer it, but otherwise I would just go to the contact page there, and you can also ask them for advice about what treatments are recommended.
Yeah, that's as you'll see on the website, that's our drinking water and groundwater protection program. And I think the best contacts are available on the website. Looks like Chris, no, I'm sorry. Yeah, Chris was the next question, saying that the tracker shows no presence of cyanobacteria right now. Is that normal? And what is the typical growth presence curve over the summer? I don't know if Lori or Peter want to try to tackle that question.
I don't mind trying this one. So I think I would say yes, it is pretty normal. Cyanobacteria blooms at this time of year are, can happen, but are not super common. They tend to get worse as the summer progresses, and increasingly, I think now we're starting to see more blooms later into the fall in September and even October. But they're really mostly a late summer phenomenon. But I don't want to be too firm in that because there can be blooms at other times of the year too.
Absolutely. Just to pick up on that, that's certainly what we've seen here. The blooms tend to start later in the season once the water has warmed up. There have been a few cases, though. I know years back we had a situation where we saw cyanobacteria on Lake Carmi at the same time we were seeing pollen. So it can be something we see, but we are seeing, certainly in terms of the life of this program, where we're now in our 19th year, blooms happening later in the season. When we first started our monitoring program around about 12 weeks, in 2019, we had a 22-week season and went to the middle of November because we saw some late season blooms in early November in parts of our waterways. And there are certain lakes, I think Valerie Dillon is here from Lake Menfor-Magog. She's been monitoring there for some time. That's a water body that's prone to some later season blooms, and that tends to be the pattern there. So it's highly variable. They can show up anytime, but they tend to definitely show up later in the season as the water warms.
Great, thanks, Lori. Looks like we have a couple questions from folks who have raised their hands. I had Peter R. as next. And you unmuted yourself for a second, and then you turned the mute back on, so we still can't hear you, but maybe, there you go.
Okay. I know you folks aren't the policy people, but I hope that you are the scientists to whom policymakers will listen. With the shift in agriculture in Vermont from the small family farms that were dispersed around the state to the increasingly larger mega farms with 1,500 and more cows, I see farmers investing in things like injection and pumping manure up to 3 miles away to make things better. What are the chances from a scientific perspective of meeting those goals at the same time as we're concentrating manure in smaller areas in the drainage basins?
Yeah, thanks, Peter, for the question. It's hard to speculate. I feel like our goal in implementing clean water projects and working with the Agency of Agriculture and DEC and the farming community is to continuously look at opportunities to reduce reduce the propensity for manure spreading, manure pumping to other parts of the catchment, to lead to surface water runoff with high concentrations of phosphorus, liquid manure, continue to reduce the spreading of manure on snow, and find solutions. Give farmers opportunities other than these practices that may not be their first choice, but are a potential solution for them. What we've seen around Lake Carmi, which is fortunately the only lake in crisis in the state, is a reduction in spreading around the lake. There is still the occasional incident. Just last summer, one of our lake advocates informed us that, hey, right across from the access area there was some spreading, and so we looked into that and found why it had happened, and that there had been some best management practices, and we've done some outreach with that farmer, and hopefully that kind of activity will be reduced in the future, so that we're not spreading manure that close to the shoreline of the one lake in crisis in the state. And I think we can build on that as a model and try to engage in similar outreach efforts and providing alternatives and solutions to farmers in some of those in the watersheds, some of those tributaries that are the most problematic, or in the watersheds of some of those sections of Lake Champlain that have the highest phosphorus concentrations. So that's I'm not answering your question directly. I think we will annually we will track our progress against TMDL targets, and I think as long as we're making progress and our estimates are sound, and we're seeing that our investments, which are considerable, are paying off, we'll feel like we're chipping away at that goal. And I think if we see the opposite happen, if we start to stall out, or if heaven forbid we actually head in the wrong direction, then I think we'll have to take a harder look at what kind of land use interventions are occurring to prevent us from achieving those targets, and have a dialogue about what additional changes are needed. So, as you mentioned, the sort of combination or what's the word I'm looking for? The gathering of larger herds into larger scale farms does prevent new challenges. And I think we'll have to adopt our practices. We're talking about agriculture as being a non-point source of pollution, but maybe there'll be pointy or non-point sources, whether it's 500 or 2,000 head of cattle concentrated in one area, and we'll have to intensify our efforts there. So I think that's the best I can do to answer now, and I'll see if anyone else wants to add anything.
Thanks for the effort.
Yeah. And we appreciate Vermonters holding us accountable and raising their concerns, and what's worked well at the Carmi example, and probably in other watersheds in the state is that partnership with local citizens, be they farmers, be they campers, and good information sharing. So please don't hesitate to raise your voice and contact us. I see that Denise Druin has her hand raised.
Hi, thank you for taking my question. And great presentation, I have to say. We really appreciate all this information. So just want to pass that on. Given that lakes are different sizes and that the bacteria, the blooms don't always, they're at one end or they're at another end of the lake, what are your recommendations for people going in the water if the bloom is not visible to them, if it's, half a mile away or what have you? Is there a safe distance or no safe distance?
So I can take a stab at that one to start. We have seen that when with our toxin sampling, that when we are not seeing a visible bloom, that we are not detecting elevated levels of toxin in the water. That said, different distances, it's one thing if you're, there's a bloom on one end of a large lake and not on another, versus if it's, right next door, that'd be closer. So I mean, there's not a great rule of thumb on the distance, but if the general water around your area that you're planning to swim in, if that general beach area and near the beach areas is looking like clean water, then I would not expect there to be elevated toxins there.
Thank you.
Great. I'm just going to check the chat again. I see we have a couple questions in the chat. I think the first one we haven't addressed is from a Bissell guest. When treating for milfoil, a large volume of plant material is killed off at once. Does this create a sudden nutrient load and perhaps add to the likelihood of blooms? So we do, as this person, as this commenter has said, we do permit lake associations and other entities to use herbicides to treat certain areas of lakes for Eurasian water milfoil, up to 40% of the lake surface area or the littoral zone surface area. And so I think the question is what happens after that, and what, at a large scale, one of the reasons why we don't allow anyone to treat 100% of a lake, besides changes to the structure of the lake bottom, which would be harmful to fish spawning and fish habitat in general. We're also always afraid of lakes shifting from a plant-dominated system to an algae-dominated system. So if you did use an herbicide and kill off invasive plants, that could wouldn't necessarily lead to a sudden nutrient load, as the question says, because the way these herbicide works is the plants disintegrate over time, over about 2 to 3 weeks after the herbicide is applied, and that doesn't lead to nutrient load, but it does lead to sort of a vacuum of plants sucking up the available nutrients, and others can come in. The whoever's opportunistic can come in, and often that can be algae. So we are concerned, and we do our herbicide permitting about creating an unintended consequence of creating that shift from a plant-dominated system to an algae-dominated system. So that's one of the reasons why we put limits on the amount of the lake that can be treated at any one time. I don't know, Peter, do you want to add anything to that?
No, I think you covered it pretty well. And I think, yeah, I wouldn't be too concerned with the direct release of nutrients from the decaying plant matter, because I think once they're bound up in those rooted plants, that takes a little while to break down to a form that the algae can use. But as Oliver said, there are a lot of different mechanisms where having a state dominated by plants can kind of reinforce itself and keep the water clearer. So we should be cautious about removing too much. And also, I would say
Oh, sorry, go ahead.
If you were to try to do some kind of mechanical removal, I think there may be more of a risk there of kicking up a lot of nutrients from the sediment into the water column, and there you could get some problems.
Yeah. You know, the herbicides that we permit today are very targeted, and so what we're hoping, best case scenario, is that the removal of the invasive plant will allow native species to return, which is sort of in line with our sort of surface water management strategy and ecosystem approach. So next question, for a lake that experiences occasional isolated blooms, what is the first thing lake residents can do to start improving water quality to mitigate? So, I mean, one thing that we always want to reinforce is when you look at the sort of the mass balance of nutrient loading to a lake, there's many sources. Obviously, there's roads, non-point sources like agricultural and logging can contribute phosphorus, stormwater runoff, you know, from roads, of course. And then lakeshore properties and impervious surface around the lake is also one of the most important sources of phosphorus to lakes. And so one thing we try to help our lakeshore property owners who are concerned about water quality in their lake with is making their properties as lake friendly as possible. And that involves allowing as much natural vegetation to return, because that vegetation is a filter for phosphorus coming in, particularly in stormwater, to slow down the flow of water across their properties, you know, by using swales or water bars or other things, and many other best management practices that are available through our voluntary Lakewise program. So I would put a plug in and say, you know, one thing, perhaps the first thing that lake residents can do to start improving water quality is to sort of start at home and start with their own properties and implement these lake-friendly practices through our Lakewise program. And there's lots of information available about that on the Vermont DEC Lakes and Ponds website. Peter, you want to add anything?
No, I got nothing, but it looks like Lori wants to chime in. So.
Yeah, I just want to reinforce that. I think that's really great. And I'm wondering if you can put it in the chat, if you can put the link in the chat box so everyone can copy it now, and also in the recording afterwards or what's going up. It would be great to have these resource links. The Lake Champlain Committee also has a lake protection pledge to just encourage people to follow simple actions. And, you know, the Lakewise program is a great program. You want to think about your whatever property you control, ensuring that you're using vegetation, as much native vegetation as you can. If you have property that goes to the shoreline, you shouldn't be mowing to the edge. You really want a diverse vegetated habitat. The more native shrubs and plants hold the soil, help develop strong and long root systems to so that water can infiltrate whatever's running onto your property. You want it absorbed rather than running off, sheeting off into our waterways. And so if you're a landowner, you have a lot of opportunity to think about the vegetation, think about creating as much permeable surface that's going to absorb the water and filter any sort of pollutants that are going on there. And, you know, we talk a lot about agriculture, certainly a significant, one of the biggest contributors and challenges in terms of nutrient loading, but acre for acre developed land produces between 3 to 6 times as much nutrient per phosphorus as agricultural land. So we can't forget that. And those of us who own property really have an opportunity to, you know, we all have a phosphorus budget, and, you know, we need to control ours.
Thanks, Lori. I think Molly had a comment. The town of Colchester has a stormwater pollution mitigation program for Colchester residents. This sounds great. You can get a free evaluation of your property, which also provides an assessment of what changes can be made to your property to reduce stormwater pollution, and then rebates to support that effort for things like rain barrels, gutter applications, trenches, rain gardens. So that sounds fantastic. So Molly, thanks for sharing that. And Molly went on to say it's a great program where we can individually work towards phosphorus reduction efforts on our own property. So I think that really echoes what Lori and I have said. Caroline had a follow-up. Any suggestion for convincing residents that the properties are a bigger or equal driver than surrounding ag? I think the point Lori just made is that on an acre for acre basis, developed parcels of land contribute more phosphorus than any other land use. You know, and I think one I mentioned this tool, which we've done on 3 lakes already, you know, led by lake associations with grants from the Basin Program and DEC QUIP program and some private consultant expertise to develop these lake watershed action plans, and we're getting those going on 6 more lakes. These plans look at all the sources of phosphorus coming into a lake and try to quantify those with either estimates or modeling or using our data, and then come up with strategies and actions to mitigate those, and then feed those actions into our watershed projects database for funding. And so these lake watershed action plans help identify the different sources and put those into context. And so for any lake that develops these, they'll see, okay, so these are the land uses in our watershed, and this is what each one of those land uses contributes. And that's often very eye-opening for folks to see, oh, I thought it was forestry, or I thought it was ag, but it's really stormwater runoff from roads or impervious surface that are the biggest culprits in terms of phosphorus loading. And the projects that come out of those plans are targeting those specific sources. So that's one other way to help people understand the respective contributions of different land uses to phosphorus. So let's see. I'm just going to quickly go back to see if anyone has their hand raised. I think we've already hit the Spencer's question and Peter R's question. So I don't believe any more hand raised. We're probably going to wrap this up in a minute. It's been a long day probably for everyone. And I think Caroline had one more question for that comment about the property of phosphorus versus agriculture. Is there any documentation you can share that we can then share with other residents? Sure. I mean, one of the slide I had earlier that showed for the Champlain TMDL, the percentage of land in different land uses, and then the sources of loads. You know, you'll see, while I'm speaking here, I'll try to find that slide, but you'll see that while the percentage of land in the Lake Champlain watershed is quite low, I think that's developed. I think it was, you know, in the somewhere in the 6 to 16%. I'm sorry that I closed the presentation, so it's taking me a second to bring it back up. Yeah, the developed land was only 4%. Developed land provided 16% of the load to Lake Champlain. And so that's one way to sort of put that figure into context. And Lori, you may have some other ideas, or Peter.
Sure. One thing I just want to note, and I think hopefully we can put in the resources when this goes up on our websites, is next week the Lake Champlain Basin Program is going to report out on the state of the lake and release their state of the lake report that will have a lot of good information and diagrams and updates on some of those statistics probably that, you know, we've shared here tonight. And so that's a really good document that will be available online, and then I think also in hard copy. So you can, you know, if you can't tune in next week, I think also feel free to email my office and we can get you a link for that.
Yeah, and I'd also say that I shared the link to the Lakewise page on our website, and that had there's a link to the science behind Lakewise, and that provides some information about the respective contribution of that, you know, 250-foot strip around lakes, you know, from where the water ends up inwards 250 feet, and the contribution of that area to the overall phosphorus budget of a lake, and that might be useful. Jeff had a question, how concerned should I be about living on the lake when the shore is in high alert for blooms, I assume, for multiple days, and should I close my windows?
Yeah, I can answer that, or I can at least respond to it. I know that's a popular question. A lot of people are concerned about that. You know, we know that cyanotoxin blooms can produce, cyanobacteria blooms can produce, odoriferous volatile compounds. So there is definitely a smell associated with some of those blooms as they die off. The smell is likely not predictive of a toxin being in the air. The chemicals that are responsible for the smell, those are different than toxins. But we really don't know to what degree that aerosolization happens. It's been shown most consistently in the ocean environment. So where you have a lot of turbulent waters and a lot of crashing ocean waves. To what extent it happens in lakes, we really don't have a lot of information. There was a paper published just recently that looked at anatoxin in the air. The researchers looked at 6 different samples in the air, and only one of the 6 samples found anatoxin, and it was the one that was taken on a very cloudy day. So we know that sunlight can break down the toxins. If you take that concentration in the air and do some sort of comparison to the level that we know is toxic, it was a very, very low amount that they did find in the air. So, it's not really answering your question, but the way that we know that cyanobacteria can make people sick is by swimming, ingesting, and drinking the water. I don't know that we know enough to say that the airborne cyanobacteria can make people sick.
Great. So I think we're out of questions, out of time, and I think we'll wrap it up here. Really just want to thank everyone for tuning in. Again, the recording of this will be up on the Agency of Natural Resources YouTube channel in the very near future. We want to thank my colleagues from Lake Champlain Committee, Vermont Department of Health, and DEC for making themselves available. Just as a funny fact, Peter Isles is joining us from Germany, and I think it's 1 o'clock in the morning where he is right now. So thanks, Peter, for sitting through this late in the night for you, and thanks everyone else for taking time on this beautiful afternoon and early evening to join us. I think all of our contacts are either in the chat or available online, and please feel free to reach out with any additional questions, and we'll continue to share our progress in understanding the trends and the health risks and our efforts to reduce blooms with Vermonters into the future. So with that, we'll wrap it up. Have a great evening, everyone.
Thank you.
Thanks, everyone.
Thanks for joining.
Thanks, guys.
Hey, Oliver.
Hey.